That's terrific, and welcome. That's Bill. I'm Steve Lucas. Some of you have met me before when doing our drone surveys and other activities. Last week we had a soil workshop, which we went into some sort of the broader aspects of what the soil does and how it behaves and responds with the clay particles, the water that we put on it, some fertiliser and the things that we change. So today we're actually going to dive into it, and there is a lot here today, but if everyone's got if everyone has at least seen what the EAL soil data results report looks like, you'll have an idea, because I'll go through one and we can have a bit of a look about the things that I find to be interesting and which we should look for in soil. So it's really, we'll start it off. So this is basically starting with a soil from out at Narrabri. And this was many years ago when we do have a standard methods for soil methods. It's Raymond and Higginson with the old ones in '94. And then these days we have Raymond and Lyon. Now these soil methods are used by every soil laboratory. So when you have a look at your soil report, you'll see that there'll be R and L and then a reference number like 15A or something like that. That's the direct reference to the soil method used to get that result. So that's just a way of being able to compare results from different soils, from different labs, and everyone's using the same method. So we're comparing apples to apples. I've put some of the first things that, the simple ones that we look at here is like the electrical conductivity of the soil, which is typically 1 part soil, 5 parts water. You shake it up and down for half an hour, and then you measure it with a electrical conductivity probe to give you an idea of the salts in that water, in that soil. Those numbers there, 0.12, anything under 0.1 is probably okay. Anything above 0.2 is probably getting a little bit moving into that saline range. And obviously, if you have an effluent irrigated woodlot, which I worked on many years ago, when you're putting nothing but salts onto the soil, it can get much higher than that. It can get up above 2. When you start getting it up above 2, for those at the lecture last week, or the presentation last week, we spoke about clay particle behaviour, the way the charge of calcium, magnesium, sodium, potassium, they all work, and they're all exchanging from the clay particles all the time. It's a dynamic equilibrium, and because of that is why we express a lot of these units in milliequivalents per 100 grams, or centimoles per kilogram. So if we look at the potassium, calcium, magnesium, sodium, they're exchangeable through a sodium chloride solution. Again, stock standard, 1 mole of sodium chloride, ammonium chloride, and you get these extracted. You get your potassium, calcium, and everything else extracted out of that. Now from that data, that gives you a balance of your cation exchange capacity, which is your effective cation exchange capacity, which is your EC/EC. Now that number there, actually all of those are quite good. Now what we're actually looking for in this is actually where we've sampled at 0 to 15 centimetres, 15 to 30, 30 to 50, 50 to 70, all the way down to 130 centimetres. So we've taken quite a big core and taken depth profiles as we've moved down. From left to right is what you're looking at. So whatever if you look at conductivity, you can see that it starts increasing by about 50 or 70 centimetres. It's at 0.42, then 0.54, and then it starts decreasing again. Well, that marries up with your A and B horizon, because that's the way it works. As the salts get leached through the soil profile, they typically make the upper horizon a lot less, have a lot less colour. And then when you get down to the deeper sections as well, that's where you can possibly have groundwater rising up and down. Again, it's going to be different for every site. But effectively that B horizon will be leached of salts as well. So there are these trends in your chemistry. So when we're looking at your crops and pastures, we're really only looking at the top 20 centimetres. That's all we're really concerned about, because that's what we want to produce. That's what we want to create more of that. And last week when Lee showed us the plugs from Rod's place, now this was at the back of Rod's shed. It's not even active paddock, but it was extremely healthy and friable and spongy. And then you could see at the extent of the roots is when it hit the clay, and it was, it was wet, moist, and it was not feeling like toothpaste, but like a good clay should. And that extra 250 millimetres of the soil that was on top of that clay is probably what Rod's produced through his activities over the last, 25 years, 22 years. So again, you look at total phosphorus. If you just look at some of these that I've highlighted in red, so organic carbon.
Sorry,
Oop.
so you can always ask a question.
Yep.
So connectivity is important because is that a measure of the exchange that's happening between plant and soil?
That gives you a relative measure of how the concentration of the salts in your soil. So if you've got a low EC, it means you'll still have a balance, but it's not salty enough to start maybe causing structural issues in your soil. It's when you start increasing, particularly sodium, in relation to others, is where you start sending the clay particles further apart because the charge is less, it saturates. Yeah. And then obviously if you get some rainfall falling onto the saline soil, it completely disperses into clay particles, and it goes down and blocks every little micropore in that top 1 centimetre of soil and won't allow water to move through it, and it'll just run straight off and typically take that dispersion layer with it. So picture that little cycle happening for 200 years in Australia. With some of the photos we showed last week, we had metres and metres of soil that had been lost in some of these areas, just through that. Soil is the result of parent material, climate, topography and time, effectively. So all the soils that we see are erosional products that so whatever the parent material was, if it's a basalt plateau or a granite, they will all determine the mineralogy of that soil. And the salts can come from groundwater. A lot of our groundwater in Australia is very salty, and if you remove the trees and you get you get this capillary movement of salty groundwater moving up. If you excavate things, like mine sites, you actually cut into that, and that's why they've got to continually pump the salt water out all the time and put it somewhere else, which is typically back up on the surface, which ends up in our creeks and streams. That's where the salts come from. And ultimately, they all end up in the ocean. And so that's our sink for the salts. But it's really, again, that dynamic equilibrium of cations dissolving, moving through the landscape, depending on where they were eroded from and sourced from. Does that sound all right, Lou? Organic C is different to your total carbon. So your organic C is sometimes described as labile carbon, which is the carbon that is most rapidly turned over in the soil with respect to bacteria and fungi, and we'll talk a lot more about that later on. So when we come down to the exchangeable cations, I went through these, you probably want an effective cation exchange capacity of 25. All of these seem to be quite high in that respect, and you can see there's like a downward trend in those. So again, what we're looking at is leaching trends through this soil profile. Really this first slide was just looking at a soil profile which we went through last week, describing the different horizons, how the soil behaves and responds to different events through, after water's fallen on it or it's been irrigated. And we summarised some of the major soils that we use in our agricultural settings in Australia. Now this is the soil results from Andrew. So we've got a range of samples. These grey columns on the right-hand side are more or less recommendations. So we talk about, if you know what type of soil you've got, whether it's a heavy soil, medium soil, light soil or sandy soil, there is ranges for, in this case, soluble calcium. So 767, oh, this is difficult, isn't it? And it says there 1150 for a clay, 750, so there's a range. So if you're interpreting that and you didn't get given those guidelines, you wouldn't know what number is really that meaningful. Well, okay, well column D, yeah, D. So that's the method reference. So on all the reports that you have, said this before. So we've got Raymond and Lyons 2011 9E2, Bray 1, Colwell, an in-house S3A Bray 2. Now this is all for phosphorus. This is just one test, or many tests to find phosphorus. So if you're looking at, you want to know how much phosphorus is in the soil, you do a total phosphorus, which would just be an acid digestion. They'd run it on, an ICP, and you would have a result, which they would express in milligrams per kilogram. So you'd have the total amount of phosphorus. The thing is with phosphorus, it's not always bioavailable when it's in the soil because it binds strongly with the clay particles. So to get that phosphorus into the plant, you need a whole heap of other processes going on, and a lot of those are biologically driven, and we'll talk about that later on. The Bray 1, Bray 2, the Colwell P, all these different phosphorus tests are exchangeable phosphorus examples. So the Bray 1 and Bray 2 might be applicable for 2 different soil types. It might be applicable for, Colwell P might be applicable for, because they're all exchangeable, all using different solutions to extract phosphorus. And they're all meant to reflect a different process that's happening in the soil between the soil and the plant. I'm picking on phosphorus because phosphorus is the limiting factor for many things. I mean, people talk about nitrogen, and you can put too much nitrogen on your pasture, and it does get into the waterways. But if you don't have any phosphorus in a high nitrogen river or creek or estuary, you won't get an algal bloom. It needs a critical level of phosphorus. So when in summer, we talk about springtime and people out putting the chicken litter on, historically in, say, down Rod's Way in the Carril catchment, there are lots of chicken farms. A lot of farmers put out the chicken litter, and we know that it's been mobilised from the land into the creeks and into the waterway because we test the water quality. Recent work with Rod and the other MEMS group down there is a number of farms have kept the cattle out of their wetland, out of the riparian areas and the mangroves and the salt marsh. And that in turn, which we'll talk about a little bit later too, is equates to the soil carbon sequestration.
Is that because they put in the wrong time of year, putting too much in?
Yeah, look, it's logistics. I mean, I've seen where, the chicken litter waits for no man. It just keeps piling up. So it's not needed all the time. So typically there'll be a timetable, someone will dump it on someone's property, and it might sit there for a couple of months in the rain and just washing off rather than being applied straight away. There's a whole heap of things, and that pile of chicken litter might have been placed just upstream from their dam, which, they're just little simple logistics. So when you test the dam water and it's full of phosphorus, you don't really have to scratch your head because you just turn around and see the pile of chicken litter there. But of course, the repercussions for that is next time it rains and the dam overflows, it mobilises all that phosphorus down to the low point.
So is the aim then to get your phosphorus down as quickly as possible?
It is, and that's the secret, or that's the mechanism in most fertilisers. So whether it's in a soluble form or when it's in a chelated form, like an osmocote type of thing, a slow release, and that's what a lot of the soil science at universities are looking at. They're trying to get the phosphate groups wedged together and then able them to release under certain soil pH conditions. And as I was saying to Trent, a lot of people go out and try and balance the calcium, put lime, but that raises the pH. And once you raise the pH, you're actually decreasing the solubility of some of these micronutrients that you really want to mobilise, or you really want to be available, because they're helping, all this electron transfer with the bacteria and the fungi and nematodes and what have you, all contributing to this dynamic equilibrium. I'll go a little bit further down here. I wanted to talk about, because this is another confusing thing. So I'll talk about the exchange. You can see we've got 3 units, and you've got 3 numbers. All of them are completely different. The centimole per kilogram number is the exchangeable calcium expressed in terms of its molecular weight and its charge. So there is a little, I think it's around 200 or something that you divide your milligram per kilogram by to give you your centimoles per kilogram. The kilogram per hectare is just extrapolated to take your milligrams per kilogram into a surface area. So which number do you use? Personally, I use this one, the 8.2, because the centimoles per kilogram are the ones that you use. If you want a calcium-magnesium ratio, it's those numbers that you put together, calcium divided by magnesium. So it's 8.2 divided by 4.8 in this case. In recent discussions, we were talking about totals, saying, well, why do we keep adding calcium, magnesium, potassium? How much is actually in there in the first place? Because remember, these are exchangeable. So we're just looking at what 1 molar ammonium chloride will extract out of the soil, whereas if you use an acid, you'd actually suck everything out of the soil. So this is the question that was asked. So we go down and we start looking at some of these totals. And you can see here, the total calcium is 2,558. Yet, it's only 1,638 milligrams per kilogram can be extracted. So there's another 800 milligrams per kilogram that is there that is just not bioavailable. So the science of all this now is we ask the question, well, what is in the soil that can help release that calcium and make it bioavailable to the plants, rather than keep throwing on more calcium because we know it's going to become available. So we're loading up the natural mineralogy of these soils, which before we came along, plenty of trees and plants and biodiversity out there. They didn't need fertiliser, they cycled themselves, they worked very well, and we have changed the landscape. So now it's up to us to come back, identify what's happening in the forest, what's happening in the pasture, and try and just make it work as best as we can. And
Can you just explain what's the difference between when you talk about soluble, something that's
Yep.
soluble,
Yep.
soluble calcium and soluble
Yep.
to exchangeable?
okay, well, the exchange solution we use for these cations is 1 molar ammonium chloride. That will give you a certain solubility, so that will extract, you know, all the cations out of solution. Water would extract a different amount. So with the ammonium chloride, we're actually loading it up with positive. We're basically saying into the soil, every site that has got calcium, magnesium, sodium or potassium on it, we want to rip them all off, but to do that we have to replace them with something. So we put in a saturated salt solution, like ammonium chloride. So ammonium chloride then binds to all those sites, takes out all the cations, and that's what we measure. If we use acid, as I said, we break every bond and do what acid does, and you'll extract everything out. But that's not how a plant works. That's not how it behaves and responds in the soil. So all of these tests are geared around trying to mimic a plant and how it would get phosphorus. Now, a plant can only get phosphorus in the form of soluble phosphorus, which is your orthophosphate. So if you apply orthophosphate to your pasture in the soluble form, rather than an insoluble form that needs to break down and decompose and slow release phosphorus, that's the seesaw that we're on at the moment when it comes to fertilising our properties. It's like, if you've ever seen those shows on the universe where you see galaxies collide and the dust of the thing, that's exactly what it's like in the soil with the hyphae. It's absolutely fascinating when you look at it under an electron microscope. Some of the ways that, it's like synapses in the brain, sparking, firing. That's how it works in the soil. That's how it should work in the soil. But what are those synapses or bacterial fungi that are firing and making it all work? We're only just opening that box now, so it's a pretty exciting time for soil scientists.
So once these cations are released,
Yep.
what's left behind?
It's exchanged. No, it will be exchanged with a hydrogen atom or another element that's sitting there, Anything with a charge will float through.
So releasing these cations doesn't replace something that's toxic?
No. It's think of it like a jar of jelly beans. It's full of black ones are hydrogen, and you'll have red, green, blue, yellow. The jar's never going to change the size, but there might be more black ones in there at certain times, which is what you want to balance, because that's going to make your soil lower pH. So the idea is to, pick the black ones out. Well, how do you do that? Well, that's an electron transfer which you can sort out with organic matter, you can sort out with bacteria, there's a whole heap of things. So all these processes, and I should bounce on, otherwise we'll be here all day. Again, you can get your base saturation calculations as percentages. And again, the percentages when you're given that in a table are different. So the takeaway message from this is look at your units, focus on the centimole per kilogram units when you can, because once you have those, you can then calculate everything else. You can just through using a spreadsheet. I argue that, you know, and I might have mentioned this last week, if you've got a rainforest, people talk about having a rainforest soil and how healthy it is, and I'd love that on my paddock, but the fact is that there's nothing in it. Everything's so efficiently cycled. The decomposers on a rainforest floor do all the work, and you know, David Attenborough's taught us anything about anything. It's how special they are. The fungi have a role, the vines have a role. They all work together to break down and cycle all these nutrients that we put on our pastures and things to make them grow better. Now, the only way to get it going like a rainforest is to fill it full of a rainforest, which makes it a bit hard to put a crop of lucerne in or have your cows in there. But the principle of how that cycling works is what we want to apply to our own soils. Now we start getting into some nitty-gritty. So this is more or less what we would see on... So when we talk about the soil microbiome, we're talking about a food chain. The whole food chain actually works. Things live, things die, things are born again. Conditions change, things shift, get wetter and drier and everything else. So if we look at this as our first step, so we've got the primary producers. So this is the trophic group that all others depend on. So we're talking about microscopic cyanobacteria, algae, bacteria, viruses, and a whole heap of other things that are a carbon mass. And keep in mind that the soil microbiome in soils around the world, yeah, I was going to say is considered to be the largest carbon sink in the world, other than algae in our oceans. So when we talk about CO2 being extracted out of the atmosphere, it's the bugs in the soil and the algae in the oceans actually doing all the heavy lifting at the moment, but we're adamant we just want to kill everything in the soil and keep going on, you know, business as usual.
Can you just explain the link between the biome, the bugs and the microbes and all that,
Yep.
and carbon? Why are they carbon?
Okay, well we did touch on this last week, because what happens is every single one of those in this food chain, so a plant, a tree will take in CO2, that CO2 will go down, and it not only goes all into the tree, it actually helps the soil microbiome populations around the roots as well. So that soil carbon, they eat it and grow bigger as well. And that's why it's not just what you see on the surface is your soil carbon, it's the roots and everything else. Now, as you go through time, these populations build and die, so you get what they call a necromass, so a mass of dead carbon in the soil, which you want to keep promoting and rolling over. And that's how soils are formed over eons. The different levels is said like the food chain. So it's like if this was the ocean and we're looking at plankton, going to zooplankton to shrimp to little fish, big fish. So that's what we're looking at here, but different cellular sort of organisms as we move through. The way that they work between each other is obviously they eat each other. When the conditions are different, like a good gang, they get more numbers and they can move into other people's spaces and things like that. We spoke last week how the formation of in the mycorrhizal and the hyphae that's formed, they produce these proteins and enzymes that can be used to help create or stabilise soil aggregates, particularly on a micropore scale. And we saw that in rod soil last week, where you could feel it. It was almost like you feel it breathing. All the little, the feedback mechanisms from the life and death, the relationship with the root, the glomalin, which is the stuff that one of the proteins that they produce. Now, within a little micropore themselves, this glomalin is used to block pores, the micropores in the soil, so it actually traps the water in there, so the biological population in that tiny, tiny little space has somewhere to live without, it's like Earth. We've got this throughout cosmic history. We're on this little planet which is only going to last for so long, and we get to enjoy it. And this is exactly what the soil's doing as well. But we don't understand it because we've really only just worried about what's on the surface. And again, this is really small, but I'm hoping you can all take a copy of this presentation home with you. So what do we actually look for in the soil? Because when I was doing my PhD 20 odd years ago, I was looking at effluent irrigated woodlot up at Branxton. And I thought, it'd be really good to get an understanding of what's actually living in this soil, what's in there. So I proposed that I'd like to go and work out, because back in the day we did a lot of DNA work in the biology section at the University of Newcastle, and I thought it'd be a good idea just to get a list, no matter how big it was, of the soil bacteria that was in my 1 hectare woodlot. Well, the response was, they laughed at me and they said, Steve, there's 1 times 10 to the 15. So that's 1 with 15 zeros next to it, organisms in 1 cubic centimetre of soil. So the next question you've got to ask, well, what the hell do we want to look for? What are the major players? What are the ones driving the processes we're looking for? And I always use the truffle story. We know a lot about truffles and the types of trees that they grow around, the places they grow. And the relationship between the fungi and the tree is well known. So we now have to take that knowledge and apply it to the hundreds of thousands of other species of vegetation out there and see what we can do. So it's a long journey, but I'm sure we can all get there. So what we're looking at here is stability. So we're looking at the stability of these populations. Do they chop and change too much? And we know they do because fires come, droughts come, and we'll talk a little bit about that later on, because that was part of the last week's talk when we looked at climate change, the soil, resilience in soil. The bacteria efficiency index. Well, if the bacteria are feeding well and the populations are increasing over time, obviously there's something moving in the right direction. Active carbon, very important, because this is the pool that drives these soil microbial populations. And I won't go too much into the numbers here, because in the metagenome report, which is one of the first metagenome reports that we've had in the area, we're still talking with Neil, who runs that show there, and a lot of the data that I'll show you today reflects a disturbed site versus a non-disturbed site. So there's no real correlation between a specific crop or a land type or anything. Aggregate stability, well, that's the Emerson aggregation test. The simple, we went through that last week, where you take a little ped of the soil, put it in some water, and you'll get a number between 1 and 8. Preferably you don't want a 7 or 8, because if you take the vegetation off that sort of soil away, it will wash. The suppressive bacteria index. Now, these are, I've got to learn up a little bit more about these, but from what I can gather, they have this relationship with the fungi as antagonists. So they're bulls butting horns in the same paddock, and they're fighting over probably a common food source or something like that. But as I said, we'll all learn together when it comes to these sorts of things over time. The Arbuscular mycorrhizal fungi, well, we do know a fair bit about that. And again, we talk about transporting phosphorus and nitrogen to the plant. So the mycorrhizal fungi are the gateway between the soil and the plant. If you don't have them, your plant won't live. We found that out with mineland rehabilitation work over the last 15 years in the Hunter Valley, where you take soil from 400 metres underground, bring it up, put it in a stockpile for a few years, and then spread it around, put some trees in it, and wonder why nothing grew in it. Well, the fact is that nothing grew in it because it didn't have any bacteria in it, didn't have anything in it at all. You could put as much fertiliser, anything you want, but it did not have the bacteria. Research at the University of Newcastle is now looking at inoculants. So if you want to plant a sclerophyll forest, a mixture of eucalypts and other native species, you can buy a test tube and inoculate your soil to make sure that you've got that there. Something quite clever. And that's the sort of thing that will happen in the future. So rather than getting a big truck turning up with tons of gypsum and fertiliser and something else, you'll probably just get a little something in the Amazon mail with a few test tubes, something that you can mix with your soil and then spread that on your own land.
The root zone knows there's plenty of bugs there. And you wash that really thoroughly into the bucket, into the bucket and add a whole lot of sugar and molasses and kelp and stuff and brew it up, keeps it air. And you know, he's putting up dates after about what he's seen under the microscope. It's absolutely turning it.
That is fascinating. Yeah.
Choose a bit of good soil, chuck it in a brew bucket and brew it up and spray it out.
Yeah, why not? Why not? Well, if you've ever, most of you have probably got composting, and I've just got my thing at home, but if you don't put anything in it for a while, you'll find a little grey fur all over it. Well, that's your mycorrhizal fungi doing its job, starting to break it down. And yeah, very important that we have that.
Hey, while we're talking about carbon,
Yes.
potential source of income in the future.
Yep.
Carbon sequestration.
Yep.
How's that going to work? Are we going to crush coal dust and bury it in the ground? Or,
Well, look, there are
plants, or how's it work?
there are a number of ways of doing that. I was saying before, there's a process called pyrolysis where you just, basically a big cooker, and you put in chunks of your trees that you've just got lying around, and you cook it at 60 or 70 degrees for 24 hours with no oxygen, and then you turn it into charcoal. Then, yeah, effectively a biochar. Yeah. And then farmers are just digging holes and then just dropping it in and then burying it again. So you can actually weigh what you're putting in, and that's how they're offsetting. So it's a good way of doing it, but
I did read somewhere though that changes the way soil behaves. There's some downside to that about how elements get exchanged.
Depending on the process, I mean, think about what happens after a forest fire. You get a release of potassium, you get, release of other things, the ground cover becomes bare. There's a whole different strategies that come through after a fire. Now, all of that is driven not only by seed banks of the vegetation, the soil type, but the bugs in the soil as well. It's the reason why same trees keep coming back in the same areas a lot of the time and others don't, because they don't have the associative bacteria that would, makes them competitive in the environment. So with soil carbon, again, where, in my view, how it fell apart many years ago when we were looking at the accounting side of it, they didn't really have an understanding of the carbon cycles in the soil. So we talk about labile carbon, which is the carbon which is the most quickly turned over. You've got other stores of carbon which take hundreds of years to turn over. I think they all have a vital role. So when it takes biochar, to me, I would add biochar at 1 or 2%, because I call them little apartment blocks for the biology, because they will live in that and they will use that carbon source to reproduce, to create more of themselves without relying solely on the plant. So it is like a little vitamin boost for the soil. So a bit of biochar, a bit of inoculation when you're planting native species on a horrible soil on a mine site has shown to work. We've got trees that are now lasting 10 years, whereas the mortality rate used to be close to 90% within 12 months for a lot of these projects.
I'm a novice in all this, but my sort of understanding what I'm interested in, I guess, is that it's the soil organic, so this biome that we're talking about, the bugs, and that's integral with pasture cover, we're talking about pasture, and the organics of that as it breaks down, it's actually not sequestering the carbon or storing carbon in the soil, so that's an organic carbon.
Yeah.
Whereas the biochar or coal, which it's been alluded to, that's not.
That's right. And until the so until the accounting, see this now with they've got the blue carbon strategy that's just come out now. So I use places down near Rod's place where they had wetlands, mangrove salt marshes that back in the day they blocked off the salt water because they wanted the land area for the livestock. What that does, and we'll throw up the slide a bit later, but when you get a population of bacteria and then you have all these external factors that can influence the shift in that population, and it might be fire, it might be flood, it might be saltwater inundation. Saltwater inundation is the one I wanted to get to because that turns back and it increases your soil organic carbon. So now that all these properties have had their wetlands fenced off from the cattle, it's probably wise to go down on the river's edge and open it back up so you allow the inundation to come back in. And then you just test the soil and you'll find that soil carbon will increase over time without even doing a thing. Now, that is something that you can get paid for at 20 bucks a tonne or something. So it's just a matter of coming up and monitoring it and measuring it. There's places at the back of Rod's place up on the ridge, which sold recently for $500,000. I think it was 100 acres. Can never build on it. Can never chop down a tree. The new owner is currently getting an income from that property. And the only thing he's got to do is go up there 2 or 3 times a year and take photos at photo points to make sure that the carbon store is still there. So as long as he doesn't touch it, he's getting paid to do it. And this is what we're getting to do. So if you can make money by leaving your trees in the ground, why wouldn't you do it?
Can I make a comment
Yeah.
in support of what you're saying? The number one process for fixing carbon and making more organic material in the soil is photosynthesis. And that brings material into the soil which feeds all the biology and all the rest of it. The biochar is it's against nature. Biochar is a human short-term way of getting some absorbing carbon in the soil. The nature's way is photosynthesis, detritus, feeding the sugars out through the roots to the bugs. The bugs bind up the nutrients so that they don't go. We talk about rainforest. In the dame tree, the water coming out of the rainforest is crystal clear and pure. Why? Because all the nutrients are bound up in the soil or in the junk that's down the bottom of the rainforest. You go next door and you go to the sugarcane plantation and they're running at double speed to try and stop the runoff from that sugar plantation poisoning the root. Because the soil nutrients there are overloaded with fertiliser and the bugs struggle. The bugs don't have to work, they don't pick up nothing to do because humans are putting nitrogen and stuff on it. The bugs don't, they go take a holiday, they go somewhere else.
Because that's on that point too, Lou, was the grain growers, the wheat people. You know, when I met them a few years back and was talking about the microbiome in, the wheat belt. And they said they tested it, but they couldn't find nothing, so they just don't test anymore. And the truth is that they've killed everything. They've got a dead soil, but they're growing wheat in it, which we make good money off, apparently. So my argument would be I'd rather see soils like, going... So this is the soil health summary for this metagen analysis, and as you can see, the overall soil health is well and truly what you'd hope. And I think Andrew made a comment that he was hoping to see more variability between these sites, because if you went to each of these sites, you'd say, there's no way in the world you'd say that they're all the same. But underneath the ground, you've got a healthy population, a healthy little cities of things which are all doing the best they can in the environment where we found them.
So that begs the question, Steve. Why is it then that you've got lovely green grass growing on this pastures which stops the land? That's the problem we're trying to solve.
Well, I think the more, again, if you look at biomass above the ground, and that's why a rainforest example is so good, because it's so dense and so thick and it's always shedding leaves, branches and cycling. It's different in a pasture where you might grow the grass to that. Rod's approach to changing the stock rotation and the time of the cattle on the property is brilliant, because you don't ever allow the grass to go so low. And we learnt that from the Charles Massey experience. His story, he talked about that last 3 or 4 years where, in hindsight, he should have taken his sheep off the property, but he didn't, because next year was going to be better. And that hope of the farmer is always going to be strong. And let's mention this to Rod, that a smart farmer these days, instead of just toughing it out and soldiering on, you really got to make the decision to say, look, I'm shutting this down and getting my livestock off this. Because if I let them take it down to bare earth, next rain event or the windstorm and my soil's gone. And you're putting yourself back by 10 years. And that's effectively what we've done in Australia with business as usual ag over, 1900s and 1800s.
But there's still a couple of contradictions that I have trouble resolving in my head. So, look at these numbers, it's pretty good. Looks all right. So
What does it mean?
how is it the A horizon is that deep?
The A horizon can be much deeper than that, depending on where you are on the hill slope. And again, this is another one of the haziness of soil science. You can take, 100 samples on a 100 hectare lot, and you would find there would be a variability range for pretty much everything you tested for. And then the next question you'd probably want to ask, well, how do I get it all the same? Because that's how I'm managing my crops, so that's what I need to know. Well, we'll never ever know that. We just have to work within, operational management and response boundaries that are leading us in a soil formation pathway, rather than the slippery slope that we've been on where we're losing it. So going back to organic matter and mulching and all these sorts of things, they're all extremely important. So whatever you're doing on your land, you have to leave 10-15% of it there to mulch back into the ground to cycle, to give the bugs the food. Because if it's pasture and you grow it down, the bugs will just start to die off. But when you want them to die off, but you want the roots to grow deeper, you want the pasture or the stuff at the top to get eaten and used up by livestock, using the cattle as an example, you want the roots to grow deep and thick, and just die there and decompose, and all of that becomes another type of carbon that can get consumed by all these primary producers. And you've got more primary producers, you end up, nematodes and worms at the other end of the scale. So we're talking about bioturbation. So you're getting aeration of the soil, which you don't get if you haven't got the living things in it. And we did that when we used to kill soils and then, till it and do all these sorts of things. So it's... Now, I won't go too much into this, but again, they are the analysis indicators that I briefly went through earlier on in this slide. So you've got your fungal consumers, aggregate stability, pH, phosphate. The trouble with, well, not the trouble, but we haven't got the full interpretation of these results yet from the laboratory. And so I wouldn't be second guessing, but as I said, what they've used is they have a library of many, many sites where they would have tested for all these bacteria and fungi, and the glucodiase sort of, again, is another protein like glomalin that's put in there to help bind soil particles together. And every single one of these soils was given a, good to excellent. Then just from the bacteria itself, so we're looking solely at the bacterial diversity. Again here there's some low ones, okay ones, on target and exceptional. Again, we're not really sure what all this means, but the balance of all these organisms is important. So the questions that Neil from the laboratory is now asking is, he would like a little bit of history on where we took the soil samples, so he can get an idea of whether it's been worked or improved or and these sorts of things over time. Because all that information is the metadata that goes in to create these libraries that we'll rely on in the future to give us an idea of, if you think of a pie chart and you've got a population, what percentage of nematodes versus bacteria versus mycorrhizal fungi do you need? And again, acidobacteria... the test is about $350 a sample, I believe, which...
So essentially just it was done with a suite of soil sampling that we did, but the important side of it was around the biology, so you're essentially trying to find active root growth effectively to make that assessment. And what we're trying to do is create some level of benchmarking around what's here, because the difficulty we have is the context that the metagenome laboratory has based on the gap. It's obviously much more intensively focused
Exactly.
A lot of information. It's not necessarily relevant to what's here. Like we've all been doing great march, but nobody's putting steel in the ground or spraying our fields or things like that in this context. But we've got to sort of extrapolate from this. How is this useful to us as far as indicators to altering practices, or what's the impact of different levels of inputs that we may put in if there's excessive, if we've got sort of benchmark levels in that soil biology, and then we see a negative decline in that as we increase chicken manure inputs or single super inputs in which has phosphorus particularly. Is it one of those areas we often see those sort of levels of traditional agriculture where the inputs have been increasing as the requirement for the output increase. And sort of here, because of the different sort of more environmentally focused operations, the high rainfall, heavy clays, we've got a lot of opportunity for input to be very limited and washed out of the system and applied to a lot of water-based aquatic systems. We're sort of looking at making sure we're effectively functioning and using what's in the system rather than just continuously working our inputs. So we understand from a practical viewpoint, there's not a lot of great content here. This is not sort of giving you something to go out and do necessarily. It's giving us this, starting this conversations about the processes and practices involved. And we are going to have to start from the point of benchmarking ourselves to our own context and being aware of that sort of process that's involved. And hoping we can get these sort of things to start the conversations and create some awareness around totals, so time these sort of things. So that's why totals tests were included in the soil tests and things like that. So we can start sort of looking at what's potentially available, or if there is no phosphorus in your soil in a totals test, well, you're not going to cycle something that doesn't exist.
If I can understand it's bad, I've never seen it advertised as to, where you get it done.
Yeah.
It's all, as I said, it's all,
There's,
you're right.
which group are you in? Group? Lanky group. KGL.
Oh no, Stroud Road, so he's up at next to Brett Cooper's. Yeah.
Yeah, right. So, I mean, I raise the point, each group had a bunch of money from last year to do stuff. If you want to get one of these things done for your place, check with your convener, the local convener, they may have some money. I don't think all the money was spent out of every group, but
What's the group up at Stroud Road with Brett Cooper? It's yours, Rod.
so, just check if there's money left in the pot, then you might want to get it done.
I guess I'm probably not going to get a very good result. My problem is I don't have enough NPK rest. It's about between 1/8 and 1/10 of what's recommended on your soil report, Andrew. So I've probably got to deal with that first before trying to understand.
Not necessarily.
No.
What the importance of it is finding your starting point. And that's probably the most important point to a lot of stuff. It's all one of the little patterns that look back and say, I've done quite well with the circle of race of cows and gone on ahead, but they may have had a very different starting point for someone who's had to do a lot more. So the starting point is actually the most important part of it. It's what we set
And
out.
just to back up what Andrew is saying, when we first moved into the property, we did a soil test way back in 2010. So then the last soil test we did, just 2020, gave us a comparison. Like we sort of said, okay, since 2010, this is what we've done, right? Then you can compare to see whether there are any benefits to what you've done or just nothing that's been wasted. I think you do have a starting point.
The point of where you're at. You might have bought an extra dairy farm that's bought stuff over for generations. Or you bought someone that bought
Don't you think they happen for? Yeah, yeah.
Bad news doesn't improve age, so you need to at least know where you're at. So that next year when you do another test, you can verify that your time, which is worth money, has been worth it.
Yeah, I understand.
Depends a lot on what you're trying to achieve. That's the first thing. If you're very clear on your goal, then you can start to find a way from where you are to where you want to be. And it doesn't necessarily come through a lot of input.
I think
Management is your biggest and strongest and probably cheapest weapon to improve your property.
I think up at your place, Michael, you're still in that discovery mode, aren't you? Where you're just feeling it out, want to have everything there to just understand it better, yeah.
You know, Dale was making the point about 20 green grass and the cows are not growing. And there's a couple of guys who referred to Joe Arbuscular and Peter Andrews talk about biodiversity in pasture. These are key. You can see these when you walk over the pasture. But it's just like us, you know. Broccoli is very nice and green, right? Eat broccoli all the time, you're not going to grow. You've got to have a balanced diet. The pastures need to be done. If you've got one monoculture in there, and particularly in poor pastures, weeds and poorer grasses will grow in poor pastures. They look good, but they're not very good. And as your pastures develop, and the clovers and the various other forms and different pieces come in, then you get a better diet for your animals. And so your animals thrive, and your pastures thrive. It's not as simple. I'll analyse this and that, it looks good, good right. Doesn't work like that.
Yes, no, well, it's true. And again, this is just the bacteria that we're looking at. If we then go in and look at the fungal diversity composition, now remember we looked at the general overall soil quality graph at the beginning. It was made up of all of these outputs. And as you can see, you've got low, okay, on target and exceptional values for the range of sites. Whole heap of different fungi there. The Mycorrhizal group is I think the genus of all of these, and as I said, they have that ability, you know, with mushrooms above ground, truffles below ground, and all these a lot of other microscopic ones. They all work together as part of this soil community. Nematodes are another one. So you can see here a lot of these are okay or on target. Again, we're still saying, what does this actually mean? I mean, if you've got a green field versus a rehabilitated mineland paddock, I'd be very surprised if we got similar numbers. And I think that's the sort of difference in disturbance where you would notice significant changes in the soil microbiome for this start. But the sort of questions I'm asking in my head from a science, research perspective is, we've already shown that you have a total and you have an exchangeable. The exchangeable is never anywhere near what's available through the mineralogy of the soil. So it has to be made bioavailable to the plant. So my simple brain says, well, what lives underground that is going to be able to transform calcium and put it into a form for the plant? And there are things out there. We found them. We have stuff that eats plastic, metals. Why can't we find something that does calcium? And I know there is one out there. Same thing for magnesium, all of these things. The whole field is called phytoremediation, and they use it in old landfill areas to plant trees and remove heavy metals from the soil. So the heavy metals get taken out of the soil and they get stored in the tree or the shrub, and then they can harvest those and take them off away from the site. So it's an exciting field. So with 1 × 10 to the 15 organisms per square centimetre, we're really just, this is not even the tip of the iceberg, but it gets the mind juices going, thinking about what you're doing on your farm, how it relates to the soil, what type of soil you've got, the chemistry you've got. Do you want to say something?
Just briefly, if you don't mind, so put some quick context to this, because just obviously, so everyone's aware of the concept behind where we're trying to get this initial sort of library of information around. There's the 2 initial DNA samples are on my own property. Then there's a low-lying region in Bulladeel. It's quite sort of flat, inundated, regularly inundated, very wet and boggy quite often. There's another area up sort of more north representing. There's a woodland area. There's another area of Topi. There's another area near Bulladeel that's sort of more of the sort of high country, just towards the sort of recently cleared. And then there's another one in the more sort of about a foot above sea level type of thing over near the... So what we're trying to do is give a bit of a cross-section across our sort of region, our context of some different areas, to then start sort of being to able an opportunity for people to extrapolate data and processes and then build something around different contexts, I suppose, because there is a lot of variability to our region. So to develop this starting point, we need to sort of have that cross-section. And the reason there's two here on my property, the biggest cock-up with my selected site for it was the fact that there was the assessment was to pick about an acre on your property that's a median representation of your place. So don't pick the boggiest spot, the hilly, just sort of find a general average and sort of locate that. And we've coupled that with a winter active pasture trial to sort of work around what are you actually growing too. We're coupling rather than just take it to a practical next step. In my property, we weren't aware, we've only been there about 5 years, and we thought it was a nice, easily accessible area near the road, short, just inside the gate, so having field days and examples, it's sort of easy of access. The downside to that is there's a circle section within the sort of pasture trial that's done exceedingly well. And then up the hill from that sort of less well. What the soil tests have shown, much higher phosphorus levels in that area, and we don't know for a fact, but we're going to assume that that's where the prior owner dumped all they took and your prior spreading. So that was literally the focus. Why wouldn't you bring a bee, double truck and dog in, dump at the gate and wheel out? So what that's shown though is from everyone given sort of equal seeds to show those sort of phosphorus levels, that increase sort of fertility and growth there. And the reason we've done 2 tests is we wanted to take a sample. One, there's an area that's going exceedingly well, and I've got a series of different trials of the same replication and going up the hill to about a 50-metre section, and then up the hill, say 30 metres, it's doing much less well. Now, it's only from here to the other end of the hall distance away, if even that. So we wanted to sort of take 2 samples from that, showing that there are a decrease in phosphorus levels, and we also wanted to look at how those increased levels may be affecting soil biology and things like that, because farm to farm, there's a lot of variables, but in a small distance like that, there's a lot less over soil type than that. So I think that's sort of the reason we double up on this one property, because we took that opportunity of unknowingly selecting that spot to extract some additional data with less variables involved. So hopefully it just gives a bit of context to what we're actually trying to do here within that.
Might point out indeed where the soil phosphorus varied by a factor of about 10, I think from 2, and there's things here up about 20. The high phosphorus area is still growing useless crap grass until we introduced our trial plots of various legumes, got spectacular results there. Still crap pastures, so it's not a one step. Increase fertility and increase the productivity of the species in the pasture mix.
So we're also with the diversity and stuff like that, and appropriate selection of species, that's sort of part of the whole picture of things as well. So, because that's why we accidentally picked this one. It's the same as the spot over there, and there's a built fence around it until we actually applied some legumes to that, and it's like, okay, that's a big difference.
I suppose
So can you just explain what relationship are you expecting to see between phosphorus and the biome results?
To be honest, I was expecting to see a much more negative effect in the biome results
So
around
high
the
phosphorus.
very focused deposition. And because I don't like chicken manure, guys, but just the context of constantly adding that without any thought to it, and what the negative effects it's going to have on your soil biology, and there was none. Our numbers from around phosphatase and those sort of things relative to that are all the same. So
yeah, the hypothesis was that if you put on too much chicken poo, you're going to burn your soil biome and kill it off.
it was 5 years ago, but one would have hoped.
We get a litre of rain a year, so.
Yeah. But the phosphorus and all that's still there, 100-fold compared to any of the other trial
Which
products.
says that binds, doesn't it? It says that binds the phosphorus.
Yeah.
Sorry.
So the phosphorus levels didn't impact the biome as much as you were expecting?
Not even remotely.
Which was interesting, because that was...
That's an interesting take from that, I felt.
Yeah.
Especially because we're trying to tie this, the biology and the soil test and together. And
Yeah, yeah. Well, see,
It is possible at a very extreme range of chicken litter there, probably sort of 50 ton chicken litter sitting there through a major rain event, sort of stay in the soil, that may have some short-term negative effect on
Yeah.
the biology left in the soil over 5, 6 years.
yeah,
From that, the soil
come back up again.
building back up and to the higher phosphorus levels there, to
Yeah.
expect those figures to build back up and sort of the positive effect on the long term.
Is there any difference in the pH of those soils you're talking about?
No.
No, they're all around 6.
And there's not a huge variation. I mean, just to look at that, particularly just looking at the colours rather than the spores, there's not a massive variation across this area. Like,
No.
they're all either kind of okay. You'd probably like to see more on target and love to see it moving up to the exceptional.
But I think the problem too, to be honest, is also within the soil tests, as Steve sort of highlighted, you've got that grey column at the end. Somebody in DLA has said, this is your ideal figures. For what, for them?
Yeah, yeah,
What are your ideal figures for you? Like, they've got to give you something so you can, oh, cool, like, I'm good at that. But it's still contextually based. And the same to this. This is based in a soil scientist based out of Gavin. You know, this is speculation, right? But you sort of got to look at sort of the context of it. He's saying, shit, yeah, if you've just mutilated your property with flowers and chemicals and shit for years, and you get these sort of figures, it's awesome. But maybe this is shit house for us.
yeah, that's. So maybe there's opportunity.
We really don't have data
No.
No.
We don't know what we should be looking for, but we've looked here and sort of seen practical results as well. We've got sort of over 2 feet high here, we go to 50 metres up there, it's over that's sort of orange factor difference. What sort of scientific testing we figured there was to quantify that, we haven't done enough testing in the whole area.
And can,
Is your goal to move this up and get a higher
Yes.
result?
And I think we can have more people involved and work out ways we can get those. And there's a greater library around
yeah.
and close to the context, high range.
So what we for every metagen analysis we get in mid-case LGA, we should be archiving that somewhere for future soil people to have a look at, particularly when we start moving into this new, DNA analyses for soil, which is, as I said, it's relatively new. And even when you talk to, there's probably 3 or 4 laboratories I know that do this work. And look, 95% of the time when you get a soil test, a chemistry soil test, the only advice that you'll get will be, addition of lime or addition of fertiliser, just to bring the balance back. That's it. So, and you don't want to be paying a soil scientist thousands and thousands of dollars to tell you that, if you can look at your own numbers and make your own decision.
And that's one of the reasons why this pasture selection trial is really important, because you're going to spend money on something, then spend money on a thing that works, because if this is supposed to be, that's the idea, and you're getting that carbon in the soil.
And speaking of balance, this was the new graph sent in last night by Neil, Andrew, which I found, to me it's a lot easier to look at, because what we're looking at is abundance and all the different classes. So you can see that, some have more Basidiomycota than others. Some are dominated more by the reddish side of it. But they're the different soils. So maybe that graph is more indicative of the differences that we witness between the sites, because there seems to be a lot more variation in that balance. So if that was a pie chart, because I work on the visuals, I'm very simple-minded, the visual there tells me I can see that, well, let's see DNA sample A and DNA sample B. Slight difference in the balance, but the breakdown, you can see all the little lines sort of, they would correlate well from from a soil microbiome sense.
Are all fungi and bugs equal?
They chop and change. As I said, if you're Lord of the Rings, 5 armies come together, that's what you're looking at. It's really that. And
Only 5?
well,
It's balance you're looking for.
that's and one will win. But then and I'll go into this in a minute, because the important thing that I look at is you know, having dealt with a lot of bacteria and viruses in water for many years, going from freshwater environments into saltwater, there's a death rate that happens, you know, it's like 18 seconds for, you know, not COVID, but we're talking noroviruses and adenoviruses that we found in the oysters. But those viruses last 18 seconds in salt water before they become inactive. But when you do the test, they send it off to New Zealand, and because they don't have DNA, it's an RNA, so it's like the COVID discussion you've heard now, you can sometimes get a false positive. They can recognise that it was there, but it doesn't tell you whether it's active or not. So these viruses, which do exist in soil as well, are all part of this diversity of different scaled organisms from the primary producers all the way up to the feeders. And this graph here, you know, instead of looking at all those green lines and everything looks okay or fine, this shows a little bit more variability, which may be related to things like soil moisture, you know, aspect and all these sorts of things. Because with bacteria, when you go through, you know, in water, they have a growth rate and a die-off rate, and I suspect the same is in the soil. And yeah, I think this will be more what we'll be looking at, because the next question that we want to ask is, why is this so important? So when we're looking at a current state, we've got this box on the left, so it represents all the different shapes and colours, represent all the different types of things that did, you know, the 1 times 10 and the 15 organisms. Very hard to put into a diagram like this. So what we want to do is we want to determine the DNA sequencing of all these organisms that, or the families of, or the groups of them that we can identify. And it can be simple things, positive gram, negative gram, all the things that you would go through different bacteria and viruses and nematodes and fungi and things as well. Learning the function of each of those individuals, their role in the underground city, how they work, their role in the army, what do they do, what is their, think of an ant or a hive mentality type of thing. These are actually a lot wilder than any ants or bees, but and they're much more dynamic, but they all have a role. And if you take one of them out of the food chain, the processes and roles will start to change, and that causes a shift in the underground soil city as well. So we have to understand, because each of these bacteria produce different enzymes and proteins which are used by other, you know, they share them around, they help the soil, they help decompose everything else that's breaking down, the roots of the plants. Going back to the rainforest, you know, this is where it's going really, really quickly. So the desired outcome is, once we have an understanding of each of the constituents, how they relate to each other, what their role is in that process, we can then start fine-tuning that cycling. And I'm talking about stuff that we haven't even invented yet. Because we haven't even discovered the bacteria. I mean, we do know we've got bacteria, as I said, that eats plastic, eats metal, breaks down petrol, biodiesel, a whole heap of things. So we're taking the same approach now, but looking at what's living in the soil, and there's just a hell of a lot more of them. So to go on, this is last slide. What we were talking about here is that little circle in the middle. It represents, you know, the hyphae, it represents some bacteria and virus and fungi and everything else. And the response to climate change will be different. So if we go up, we have elevated CO2. So what we're saying is the atmosphere becomes more concentrated with CO2, which it is. But what that actually does is that releases soil organic carbon, because the soil's getting hotter. It's like baking it away, as Rod would describe it as. So it actually contributes more CO2 if you've got an increasing CO2 environment. Fire, again, actually starts absorbing the organic carbon back into the soil because of the processes that needs to require, because everything's charred. You know, it makes sense that if the whole thing's biochar and you've brought it to the ground and it stays in situ, that will become part of the new soil carbon bank as you move on. The one I said I find quite exciting is the seawater intrusion, because we're in a, you know, we have sea level rise. There's many wetlands around the world, Lou, as you know, and a lot of them are held back with tidal walls and things like this to maintain the ecosystem that they've got. Now, if the salt water comes up and starts moving into these systems, it will change them. There's no two ways about it. But in terms of soil organic carbon, it will actually start drawing it back into the soil again. Increased precipitation. You know, not much drought. Obviously, again, the baking approach, more drought, less ground cover, baking the soil, capillary rise of water, CO2 gone, soil organic carbon gone. Permafrost thaw, we don't have to worry too much about this in Australia, but it's something that you could probably use in Siberia or Mongolia or up in the Scandinavian regions as a soil carbon sequestration accounting thing in the future, because it does hold a lot of organic carbon over the winter times. And the thaw causes different processes and moves it, of course. Increased temperature, I think I've pretty much got through them, but we went through this last week in our presentation, which was looking at those, the impacts of climate change and soil resilience. So how do you create a soil that can potentially cope with every single one of those externalities that changes that population diversity in the first place? So that's all I've got for the moment, and more than happy, after Andrew's had a talk about what he's doing on farm, and we'll be around for a little while and if you've got any specific questions. But keep in mind that all the reports that I've seen so far today, the recommendations, if you've got any, is going to be gypsum and chicken litter or some sort of appliance, and that's to bring up the balance to whatever that balance is. I'm not sure, but it's more than likely going to be related. Well, you might be able to tell us more about the precision, grass pastures and how that works. And Rod's got his system, and some of the ones I've seen have been, seem pretty effective.